So it's great to, as a second speaker, to introduce Dr. Michael Letko from Washington State University. You can see what he's going to be talking about. Michael started his postgraduate career in the Department of Microbiology at Mount Sinai in New York City, a postdoctoral fellowship at NIH's Rocky Mountain Labs in Montana, which have a really long history of doing very exciting, impactful work, to his studies at Washington State University, where he's in the Paul Allen School of Global Public Health, where he has a faculty position. So I leave Michael to talk to you now, rather than me try and clumsily introduce the science of his talk. But this is going to be a great pair to the really interesting presentation that we've just seen from Dr. Plowright. Over to you, Michael. Thank you very much.
Great. Thank you so much for having me. I pasted a link in the chat for the preprint that describes a lot of the work that I'll talk about today. So yeah, this work, today I'm going to mostly focus on MERS coronavirus and the family of MERS coronaviruses. So these viruses were first discovered in first discovered in humans in 2012. And it was found that the virus basically repeatedly transmits from dromedary camels into humans. And it continuously does this, which leads to a low-level kind of recurring outbreak in the region. Thankfully, it doesn't spread between human very well because it has quite an alarming mortality rate in the 2,700 or 2,800 cases that we've now had reported. We're having about a 36% mortality rate. And since this virus was discovered in camels, many viral ecology groups have gone out and discovered additional morbicoviruses in an arrangement of mammals, from bats, hedgehogs, and pangolins across Asia, Africa, Europe, and the Middle East. So there are thousands of related viruses to MERS coronavirus that we collectively refer to as the morbicoviruses. Unfortunately, only about 6 of these viruses has been studied since 2012, like in the laboratory. We have a lot of sequencing data, but not a lot of laboratory data. So that leaves the question of, are these viruses a zoonotic risk to humans? Largely unknown. So why is this though? Well, isolating viruses, especially ones we don't really understand very well, is quite difficult. So no cell line that we have in the lab is actually capable of expressing every receptor that we need for every virus. There aren't cell lines available from every tissue of every animal. So if it's a species-specific interaction, the chances of us getting it to work on our human cell lines is not maybe feasible. Some viruses may require other extracellular factors that we're only beginning now to understand. For example, sapoviruses, which are found in the gastrointestinal tract, require bile acid in addition to having all the right other host cell stuff. Receptors we know are not the only blocks, so there's other factors, innate immune system factors that can interfere with viral replication if the virus is not tuned for that cell system. We also have limited sample sizes, oftentimes from field studies. You don't get tons and tons of sample from a bat to be able to try different isolation techniques, so that limits what you can do with them. There may be other microbes present in the sample, which can interfere with our ability to isolate new viruses. And beyond that, then we have sample storage and other logistical constraints, shipping permits, handling regulations. All of these things basically kind of make isolating viruses a much harder task. So needless to say, sequencing is easier. So that means that if we need to study these viruses, we have to rely on oftentimes we have to rely on synthetic biology and basically producing something from nothing. And so luckily, synthetic biology has caught up quite a lot over the few years that we've had it. So oligonucleotide synthesis is a very common routine laboratory method where basically companies, you can even outsource this, companies will make primers. It's a routine process. Unfortunately, length dictates price. So our primers, which are maybe 60 base pairs or 60 nucleotides long at the most, only $15 or $20 US. But if we expand this to larger sequences, it quickly becomes a very expensive challenge. And if you're studying novel coronaviruses that you don't have isolates of, then making one of these genomes is quite expensive. So the 30 kb coronavirus genome will cost about $15,000 It can take several months just to have a company synthesize it, if they can synthesize it, and then it takes months usually to recover the virus. So if we narrow this question down to just a critical region of the viral genome that we want to study, let's say the spike gene, this is still several thousand base pairs, which is still, if you want to study a lot of these viruses, spending several $1,000s per sequence becomes kind of expensive. So my work really started as a postdoc looking at this question and saying, how can we study more of these viruses for less? So, what is the smallest region that we can narrow down our study to and still be able to gain some information about a virus's ability to interact with human cells? And so at the time we started this work with the sarbecoviruses, there are hundreds of these viruses, and only a few of them had been characterized, but we had a fair amount of structural and mechanistic data for SARS-1, which, this is slightly before SARS-2. And because we had some virus data in the literature, we could go back and kind of compare whatever we were finding with our platform with the data. So to get into this approach, basically, we focus on the spike gene. So entry is a major species barrier for the coronaviruses. Oftentimes, we can get these coronaviruses to replicate in cells from really diverse species background, as long as we provide them a receptor. So just focusing on cell entry then, we look at the spike gene of the coronavirus, and more specifically, the sarbecoviruses have a very similar kind of arrangement of their spike gene. It's two components, and then the part that actually physically touches the cell is called the receptor binding domain. It's a single contiguous domain located in the genetic sequence. So SARS is a small few hundred base pairs fragment. And we found that it actually is very conserved across all the sarbecoviruses. They all basically have this gene, this receptor binding domain in the exact same spot. So then the question is, can we just replace the RBD from one virus with that of another and use that as a way to test the receptor binding domain function without having to make a whole spike every time? So this really gets at the heart of what my lab is interested in, which is, we call it functional viromics, which is basically describes scalable laboratory methods to study multiple uncharacterized viruses in parallel. And this work actually went really well for the sarbecoviruses. We were able to produce a system where we could replace the SARS-1 spike receptor binding domain with that from SARS-2 or any other sarbecovirus. We were able to show that the sarbecoviruses assort into a number of clades based off of their receptor binding domain. We were able to characterize the entry factor, ACE2 for SARS-2, just 12 days after the genome was published online. So if you go back on bioRxiv, we actually have the first preprint with laboratory data on SARS-2, which was pretty exciting, which really is also mostly a testament to the speed and efficiency of this system, because we can really go from genotype to phenotype in a very short amount of time with this kind of streamlined, optimized approach. When I started my lab at WSU, I partnered with our computational and engineering department, and we were able to basically perform a pilot study trying to predict entry phenotypes from sequence alone. And this was all based off of the data that we had aggregated in the first paper. So as we learn more about more of these viruses, can we learn something and extrapolate trends from the data? I partnered with the Wuhan Institute of Virology over the past few years, and we've been able to kind of dig on some of these other groups of coronaviruses that we identified as having zoonotic risk. We were able to actually make and test reverse genetics clones that could replicate in the absence of ACE2. We developed methods to isolate ACE2 independent sarbecoviruses and actually pulled the first one of these viruses out of a bat fecal sample just last year. And now that we have this tool and more groups continuously discover sarbecoviruses, we can plug these new sequences into our system and understand their risk. So I think one of our better examples of this was with a Russian sarbecovirus that was discovered just in 2021. And we were able to characterize that it had a entry, it could use human ACE2 within a year. And then the next year, we were able to partner with a different group developing antivirals who was able to then show that their antiviral could inhibit the Russian sarbecovirus. So I thought this was a really kind of cool concept, going from sequence data to potential therapeutic intervention within just a year or two of discovering a completely new viral sequence. Currently, we're studying some of the entry factors. We found viruses that could infect cells in the absence of their host cell receptor, ACE2. So we're basically digging on this group of sarbecoviruses and are trying to identify receptors and entry factors for them. But today's talk is to really focus on the mirbicoviruses. So this has been where we've really been expanding our focus and kind of moving away from the sarbecoviruses a little bit and into the next group that we're interested in. So we've now developed a system, we call it Mirbicotype, and it's very similar design to our SARS platform. Basically, we take the MERS coronavirus spike gene, we remove the receptor binding domain, we replace a synthetic receptor binding domain in place. We can then use this synthetic chimeric construct to recover viral pseudotyped particles that contain the chimeric spike on the outside. And then when we infect cells with these chimeric spike pseudotypes, if there is entry, we can measure that with luciferase and GFP signal. So I started this work actually in 2018, at the same time that I started the SARS work. And I have to say the sarbecoviruses were a lot easier. And the reason for that is the incredible diversity we find in the mirbicoviruses. So unlike the sarbecoviruses, which are very homogeneous, relatively speaking, the mirbicoviruses can be quite different from each other. And at the spike, within the spike gene and the receptor binding domain, it can be sometimes as low as 65% similar to each other. So when you're looking for conserved motifs, it becomes very difficult. For all the sarbecoviruses, there are large stretches of amino acids that are highly conserved in all sarbecoviruses that flank the receptor binding domain. But as you can see from this little mini alignment, there isn't really the same kind of conserved sequence stretches in the mirbicoviruses. However, there are, after staring at the sequences for quite a while, I found that there are some conserved glycines. And these flank the receptor binding domain. And they're found in every mirbicovirus. So if we take a look at these structurally, this is the MERS spike trimer bound to DPP4. These glycines are located at the base of the receptor binding domain. So the idea is that we can maybe call these flexible glycine points a place where we could try to replace the receptor binding domain. So if we now have this concept in mind, how do we actually test that we can replace the receptor binding domain? And as we were figuring this out, this amazing study came out from Huan Yan, Wuhan University, and he showed that basically some mirbicoviruses, in contrast to the paradigm that mirbicoviruses use DPP4, they showed that actually some bat mirbicoviruses found in African bats can use ACE2. So with this in mind, we wondered, okay, if we have some mirbicoviruses that use a completely different receptor, can we use these glycines to delineate the receptor exchange, the RBD exchange, to change receptor tropism? So I made several different constructs. I'm showing here a little schematic overview. We've got a full-length MERS spike. We have some full-length HKU4 spikes. These are bat mirbicoviruses that have been shown to use DPP4. And then I have a version of the MERS spike where I've replaced its receptor binding domain with an HKU4 RBD to see if I can retain DPP4 use. And then I've also replaced it with the RBD from one of these bat mirbicoviruses that uses ACE2 called PDF2180. And then because I like to always see if we can get a little cheaper with our question, I tried slightly smaller versions of the receptor binding domain to see if we could reduce the amount of size of sequence that we had to have synthesized. So on 293 T cells, which are not very permissive to these viruses, we can see actually that, that nothing really gets in. If we add DPP4, we can see that MERS gets into these viruses quite well. If we add ACE2, we can see that our chimeric PDF2180 spike gets in. Importantly, though, it's only the big version. None of the smaller ones actually work. So this seems to be the minimal region that we need for this to work. So if we add trypsin, which is kind of one of our tricks we use in the lab to basically amplify our signal. So trypsin helps facilitate viral entry post-binding. And when we add it into the mix during infection, it helps the virus that has maybe low interaction with a receptor, enter those cells more. Importantly, it is still receptor specific. So while it increases our ACE2 dependent entry, we still don't see this in the absence of receptor. So we still need the receptor for this to work, but this just kind of kicks up the signal a little bit more. So we can see some of these lower affinity interactions really come up when we add in trypsin. So at least this tells us that these highly conserved glycine residues seem to flank what we'd call the functional morbicovirus receptor binding domain. So with this tool in hand, we went to GenBank and we started digging out MERS sequences. So if you look for morbicoviruses, you get close to 2,000 entries. However, a lot of these are only partial genomes. So if we look for genomes with a full spike, we can really parse it down to about 800. If we pull out all of the viruses that were found in humans and camels, which tend to be just various isolates of just MERS coronavirus, because as soon as we found MERS in humans, we did a lot of sequencing. And then as we found MERS in camels, there was a lot more sequencing. So if we just parse out MERS sequences, so we get to like the bat morbicoviruses and the truly uncharacterized ones, we're left with about 65 different viruses. And then if we look at those, so there's about 65 spikes, but if we look at their receptor binding domains, there's only about 34 or 35 unique receptor binding domain sequences that are published. They'll put an asterisk here because this is kind of continuously ticking up as groups continuously find these viruses. But, as a snapshot, when we did this study, when we first made this panel, these were about the 34 different viruses. And then we have MERS down here as a positive control. So looking at the receptor binding domain, we made a phylogenetic tree, and we could see that these viruses seem to cluster into what appeared to be about 4 different clades. And this is based off of, the sequence data predominantly. So if we look, basically, all of the clade 1 receptor binding domains have this similar kind of indel pattern with relationship to the other morbicovirus RBDs. So we can kind of look just visually at a sequence at the gaps and say, okay, this looks like a clade 4 or a 3 or a 2. And as you'll see, this relates to their function as well. So we made a large panel of viral pseudotypes, and we placed them on cells. And we are basically now looking at this large panel of 35 different viruses and how they infect different cells. So these are 293 T cells. It's a human cell line without a receptor. We don't really get a whole lot of entry except with MERS coronavirus, which gives us a little bit of low-level entry with this cell line. If we add in some human cell receptors or human coronavirus receptors, we can see we can start to reproduce the phenotypes that we expect. So clade 1 viruses, as has been shown, many of these viruses, they use DPP4. Surprisingly, really none of the other morbicoviruses are capable of using the human DPP4. If we add in human ACE2, we can get the 2 viruses that were characterized previously by Huan Yan's group to show ACE2 usage. And we don't really see anything but our positive control 229E using APN. If we add trypsin, we increase this signal a little bit. And we can see that some of these other viruses kind of give us some low-level signal. But really what stands out are the clade 1s and the clade 3s. When we added in some different animal species orthologs of these receptors, so now I'm showing Pipistrellus abramus ACE2. This is from the Japanese house bat. Japanese house bat is the animal that many of these HKU5 viruses were sequenced in. We also have the European hedgehog ACE2 and DPP4, which the European hedgehog is the host that many of the European hedgehog coronaviruses were found in. So we can see really that when we add in Pipistrellus abramus ACE2, though, we really saw this clear and obvious entry, though, that the HKU5 receptor binding domains could enter with this. We're not really sure with clade 4. We get some entry, but nothing was ever really consistent for one single receptor. So we're still playing around with that a little bit. So to come back to this ACE2 with HKU5, this was a new finding. So to really test this in a different way, we tried a different cell line. These are baby hamster kidney cells or BHK cells. They're generally not permissive to the morbicoviruses or sarbecoviruses. So here we can see that our MERS doesn't give us any entry like we saw with the 293Ts. So this really cleaned up our entry signal, and we can see basically that it is just this one group of viruses that uses this specific ACE2, and it's very species matched. So that was transfection and chimeric spikes. What about some other things to kind of really dig on this? So we paired up with Ralph Barrick's lab at University of North Carolina. They have specialty in replication competent coronaviruses. They have an HKU5 molecular clone in their lab that they've been trying to grow for years unsuccessfully. So we sent them our cell lines with ACE2 from the Pipistrellus abramus bats, and we could see actually their virus replicate quite nicely on these cells. So looking at the receptor use, then it seems like the mirbicoviruses kind of share a similar story as what we've found with the sarbecoviruses, and that there are these patterns of indels and sequence and entry phenotype that kind of delineate and define the functional clade for the receptor binding domain. Importantly, though, it's not really driven by the geographic origin. So you can see that like some of these viruses are found in fairly disparate regions, but have similar clade distribution. So I think that probably says more about our sampling, about being a little bit biased, but it also suggests that we're likely to find more morbicoviruses as we look more. So, okay, so we know that HKU5 can potentially use bat ACE2. What about other ACE2s? And so to look at this, we kind of turn our attention to the ACE2 using morbicoviruses. And so clade 3, these are the two viruses that were characterized in the earlier study. They contact ACE2 through a series of contacts, but I think what made this question such a challenge for predicting is that none of these residues is really conserved in the HKU5 viruses. And to further complicate things, the HKU5 viruses have a lot of polymorphisms in their receptor binding domain, including this group that has a deletion in one of these loops. So if we take a look at the structure for this, this is the PDF 2180 co-structure that was published with its specific bat ACE2, which is from Pipistrellus bats. And we can see the two loops contact the ACE2. Here I'm showing one of the, we call this a two-loop HKU5. This one, if we go back, has both loops kind of fully intact. We can see both of these loops make a contact with the ACE2. If we shorten or we use one of the other viruses that has a shortened loop, we can see that this may not actually contact the ACE2 as efficiently. So focusing on these two loops, we have two different amino acids that we mutated several different ways, and then we compared entry on different ACE2s. And so what we found was that ACE2, this is from Pipistrellus abramus bats, and we have two different cell lines expressing it. So we have hamster cells and human cells expressing the bat receptor. And we can see that basically in comparison to the wild type, which is shown in white, all of these other mutations at both loops in both viruses reduce the ability of that virus to infect or use ACE2 from the Pipistrellus bats. However, if we then test it with the Homo sapiens or the human ACE2 in the same cell line background, we can see that the virus that has the larger loop is actually potentially able to gain an increase in its binding ability with human ACE2. So, and in contrast, the virus that has the smaller loop, we don't see this same kind of consistent increase in entry. So this suggests that maybe the second loop or the larger loop allows these viruses more of a flexible surface for adapting to a new receptor. So real structure and deep mutagenesis are needed, and we're kind of working on that as well. So we've paired up with Pamela Bjorkman at Caltech, who specializes in cryo-EM. And while we're still working on a co-structure of ACE2 with HKU5 trimer, we do have the trimer alone, which this is the first HKU5 trimer that's been published. And what we're seeing is that that the receptor binding domains are basically all closed down in a lot kind of inaccessible conformation, which is what has been reported for many of the other bat gastrointestinal viruses that we have structures for their spike proteins. So it might be just a conserved feature of these viruses, but this is something we're still working on. So HKU4 and HKU5 were discovered almost 20 years ago, 6 years before the MERS coronavirus outbreak, and they just sat in sequencing databases. And then after MERS emerged, several groups were able to find that one of these bat viruses at least used the same receptor as MERS was at the same time. HKU5, on the other hand, this group was not able to find a receptor or really the entry phenotype. So but importantly, they really largely missed the interaction because they only tested bat and human DPP4. Then in 2019, a group then studied the reverse genetics molecular clone, and they found that trypsin could get these viruses to replicate. But again, without the receptor, they couldn't go much further. And this was largely because they were still focused on just a DPP4 and maybe trypsin now. Then, as we started to learn that ACE2 might be involved, this group tested a bunch of different ACE2s from 44 different bat species, including the closely related pipistrellus bat, but not Pipistrellus abramus bat. So what we've learned is that these interactions can be very species specific. So just to kind of go through the major points, we show that the RBD is flanked by some conserved glycine residues. We were able to characterize all the morbicovirus RBDs into about 4 different clades based off of indels and entry phenotype. Some data I didn't show here was with some consensus sequence, but we show that in general, clade 1 viruses use DPP4, clade 3 use ACE2, clade 2 use bat ACE2, but only specifically from their own host species. And the clade 4 viruses use something that we're not sure of yet. And we show different viruses in HKU5 that can influence its entry. And there's a paper that was just published yesterday, a really exciting paper. I encourage everybody to take a look at this one. They basically sequenced dead animals on fur farms across China at the interface between the fur farms and the wildlife surrounding them. And they found HKU5 viruses now outside of bats and in mink on a fur farm. So even if we think these viruses may have a species-specific interaction, those likely can be easily overcome. So future directions, we're working more on the structure with the whole spike trimer. We're working on deep scanning mutagenesis with Tyler Starr. We also have several new findings from this that are only just now coming online for us. So hopefully I'll be able to talk more about those at the Keystone Positive Strand later this year. And so with that, actually, we'll just move on and I'll thank my collaborators. And if we still have time for questions, I will do those as well. Thank you.
Wow. Michael, thank you. That's a huge body of work you've just presented to us. Fantastic. It's yeah, the implications of that and the possibilities of it are enormous. Thank you very much. Yeah. So I'd like to open it, things out now to the audience for questions. We are a little bit getting towards the top of the hour.
Yeah, sorry.
But please do, of course, feel free to leave at any time. At least people, if you have to move on to the next one. But let's have a few minutes for questions for Michael, if there are any out there. Michael, can I ask you a question which isn't really the stuff that you've been researching, but I wonder whether you could make any kind of comment about recombination, because these I mean, obviously, the amazing insight you're getting into the species specificity of these different virus families, clades. I mean, it's really, really important implications from that. But if some of these coronaviruses find it quite easy to recombine with others, then
right.
What does that mean in relation to? Does that happen often enough for this to be a significant risk in relation to interspecific transmission, or
yeah. So there's a study from Zhengli Shi's group in 2019 where they found or 2017 they sequenced a cave of bats in China. and they found just dozens of these sarbecoviruses, and they're all, different from each other, but they're all being found in this population at the same time.
Yeah.
So I think, the more that we look at those viruses, we find basically the bits and pieces of the SARS-1 genome. So I think that, recombination is a continual process in at least some of these viruses and at least some of these bats or some of these hosts. And that I kind of just describe it as looking at a river. You get an idea for the river, but you don't actually, anytime you reach in, you're going to get something a little bit different than what you did before. So, but I think the power of this approach is, we see recombination at the S1, S2 junction, but usually there's not a ton within the RBD itself. So I think by focusing on just this little short stretch, that helps us kind of, regardless of what recombines, hopefully we still have a good snapshot of entry.
Yeah, thank you. That makes a lot of sense. Michael, there's a question from Nazia Thakur in the text. Let me read it out for people who aren't on it or on their phones. How functionally relevant do you think adding exogenous trypsin is to your entry studies, or do you think it gives an exaggerated phenotype in some cases?
So I think trypsin is important because a lot of these viruses, especially the bat ones, we find in the gastrointestinal tract, in gastrointestinal samples. The GI tract is loaded with external proteases, including trypsin. So I don't think it's too far-fetched to include external protease as a factor. With that said, it does increase our entry, and our system is a little exaggerated because we use, we call it an optimized spike backbone. A MERS spike works really well in many of these cell lines. We know that it attaches really well, and we know that the cell proteases can process the spike quite well. So yeah, I think we do get an artificial kind of amplification of the signal, but that's in a way the point, to see even if those low-level interactions can be measured. That tells us something, because I think any possible amount of compatibility with a spike and receptor can then be overcome eventually with mutation. So those little interactions are also important to us.
Thank you. Michael, Raina, thank you both very much for what's been a really great pair of talks this afternoon. It's great that you've been able to spend the time to come talk to us. Thank you both very much indeed. I'm sure that, although I haven't asked them, I'm sure that both of them will be pleased to try and answer any questions that you might follow up by email. But I'm not making a commitment for them, but it's worth the ask if you've got something that really burning comes up to you in 5 minutes' time. So at that, then I'm going to say the seminar is closed. Thank you all very much for coming and making it a good afternoon. I've really enjoyed it. Thank you both very much.